Well, hello, everyone. We are live. Thank you for joining us. Welcome to our academic webinar series featuring Professor Samuel Egieyeh. First off, I'd like to mention that we will have a Q&A at the end of the presentation, so you can leave any questions for Dr. Egieyeh in the chat bar, and we'll try to answer as many as we can at the end. My name is Chris. I'm the scientific support lead here at DrugBank, and I'll be the host of this session. Each year, millions of scientific journals are published that have the potential to deliver really much better health outcomes. DrugBank works daily to ensure that this huge amount of data can be used to accelerate research and change lives. As a result, we see firsthand the amazing work that researchers around the world are doing using DrugBank and other sources. And this webinar series is meant to highlight this work and to give the researchers a platform to share their discoveries. So today we're sitting down with Professor Samuel Egieyeh to discuss his work in computational methods to identify repurposing opportunities during the COVID-19 pandemic. I'm going to hand the microphone off to Dr. Egieyeh just to give us a brief intro.
Thank you, Chris. What an opportunity to speak on this platform. My name is, like I said, Professor Samuel Egieyeh. I'm an associate professor at the University of the Western Cape in the lovely Cape Town city in South Africa. My background is pharmacy. I started the first degree in pharmacy, second degree in pharmacy, then I did a PhD in bioinformatics. And since then, I've been doing drug discovery, trying to apply various analytics to various databases, including DrugBank, to identify drugs for infectious and non-infectious diseases. Thank you.
Excellent. Thank you for that introduction. Okay, so we're going to start the video right away here to show the presentation. And remember, please put any and all questions you have into the chat bar, and we'll get to them after the presentation.
Good morning, good afternoon, or good evening, wherever you are. A warm welcome from Cape Town. It's a privilege for me to speak and present the results from our research on this platform. Today, I'll be talking about a computational drug repurposing strategy that predicted peptide-based drugs that can potentially inhibit interaction of SARS CoV2 spike protein with its human target. This is a brief outline of what I'll be talking about. So this is the University of the Western Cape in the city, beautiful city of Cape Town, and it's located on the Cape Flats of the Western Cape, of the Cape Town. And here is where I'm speaking to you from, the beautiful life science building, and opposite it is the School of Pharmacy. The DrugBank, I came across this database a couple of years ago, quite a long time ago. I was using in the teaching of pharmacology, and during my PhD, I think I got a bit more interested in it and how I can use it to in my research, as it were. So, interestingly, I just did a quick profiling of the DrugBank and looking at what the chemical space looks like in terms of a fragment fingerprints of the compounds within the database. And what we saw was that quite distinct clusters were formed. And if we zoom in on one of the clusters, we could see the differences in terms of structural similarity, measuring structural similarity with fragment fingerprints. We could see diverse clusters, and the key here shows from red to green, red being not similar at all, and of course one being very similar. So this particular cluster, you could see how similar it is to other clusters and other compounds within the database. So generally, we see a very diverse compound set within this space. Secondly, we set out to look at some essential descriptors, particularly looking at functional groups that are predominant within the database. And when we did a box plot of these various groups, what we observed was the amides. There seems to be quite a lot of them within the database, as you can see from the outliers in the box plot. But what I've seen, my experience has been that within the FDA approved drugs, and this aligned with what other people have observed, there seems to be a lot of nitrogen containing heterocyclic moieties compared to what you see in natural product databases, where you have oxygen containing heterocyclic moieties. The other thing that we also looked at in terms of describing the drug bank was the number of hydrogen acceptors and donors, and we see slightly more hydrogen acceptors. And of course, we know that hydrogen bonds play a crucial role in determining the specificity of ligand binding or interaction with macromolecules. And finally, we looked at the surface area in terms of the polar surface area, the van der Waals surface area, and of course the total surface area, and comparing the van der Waals surface area to the polar surface area, we see a slight difference with the van der Waals surface area being a bit more, and of course this van der Waals surface area is being implicated in the interaction of these compounds with macromolecules. The polar surface area also has been implicated in the disposition of most drugs, basically looking at the absorption or as a determinant of the absorption of most drugs. Okay, going to the thick of things, if you're in drug discovery, you will have seen this chart or this version of this figure. And basically what it communicates is the high cost of drug discovery and development, and the high attrition rates that follows that. And of course, some of the things, factors that have been identified as determinant of these high attrition rates, toxicity, lack of efficacy in some of the drugs that compounds that we started with, or the fact that you just don't have innovative compounds coming through the pipelines. So if you think about this, drug discovery and development end-to-end within this value chain is more or less very difficult within low-income countries. So drug repurposing, for me, it's kind of comes into that space to try to help countries that cannot afford the drug discovery and development process. So the word repurposing, I try to define what it is the use of something for a purpose other than its original intended use. Now, we are taking advantage of the fact that we know that most drugs have off-target. Although we design drugs to be as specific as possible to a particular target, but they also have effect on other targets. And that, of course, led to what we commonly know as side effects. Now, once we know that, we can begin to think about, can we use a side effect as the main indication for a particular drug? And that essentially is what drug repurposing set out to do. So it gives us a shorter time to development because these drugs have already gone through the development pipeline, the clinical development phase, and even the approval phase. So they satisfy the regulatory requirements, as it were. So the cost of repurposing is a lot cheaper than designing a drug, ab initio design of a drug for a particular disease. A very good example, especially related to my discourse today, is metformin. In a clinical trial that was reported in Lancet, it was shown that outpatient treatment with metformin reduced long COVID incidence by about 41%. And so this is very good because metformin is globally available, doesn't cost so much, and it has a very good safety profile. Okay, so diving into COVID-19 and SARS CoV2, which is the causative virus. Many of us do not need introduction into this. We've all felt the symptoms. Most of us will have felt the symptoms of COVID-19 caused by this virus. I'm a witness of that because I've had it twice. So I'm not going to dwell on it because a lot of us know what the symptoms and the impact of this particular disease was. I'm not sure it's the level it is right now. But in terms of how it came about, it's been reported that the virus binds to the human angiotensin converting enzyme 2, the HACE2, and through that interaction, finds its way into the cell. Now, I'm not going to get into the details of HACE2. It's been implicated in so many other diseases when it's been discovered recently. But majorly just to mention that it's a transmembrane protein with the N-terminus catalytic site on the extracellular portion and the C-terminus within this cytoplasmic domain that has, some regulatory sites within it that control through the level of expression of this particular enzyme on the membrane. But most importantly, what happens is that there's a particular enzyme that cleaves this HACE2 and releases a peptide that binds to angiotensin 2 that was, if you think about the angiotensin, what they call the RAS system, I'm not going to talk about now. The final output of that system is angiotensin 2 that raises blood pressure and also has an inflammatory effect within the body. But when angiotensin HACE2 catalytic site is removed, it binds to angiotensin 2 and converts it to angiotensin 1 to 7. And angiotensin 2 and angiotensin 1 to 7 have opposite effects within the body. As you can see here, one raises the blood pressure while angiotensin 1 to 7 lowers the blood pressure. But my focus is looking at the HACE2 itself. One of the questions that were asked at the beginning was that, does the virus bind more to the cleaved HACE2 or the intact HACE2? And I think the conclusion from that, without getting too much into the details, was that it seems to bind to the intact HACE2. We can talk about that during the question section. On this, I showed you the details of the interaction on a very good video that I got from YouTube, referenced here. You can see that how the virus approaches HACE2, interact with the receptor binding domain of the spike protein, and through a series of conformational changes, the virus now get access into the cells and is able to ingress into that particular cell. So this is what we just need. We know there's a receptor binding domain and there's an interacting surface on the HACE2 for that initial contact, and that is what we set out to disrupt. So in terms of methodology, let me just go back one step. Right. Okay, so we set out first to calculate the average structure of the structure of the spike protein receptor binding domain and the HACE2 that we found on protein data bank. I'll talk more to why we have to do that. And secondly, we conducted a molecular docking using the DrugBank as our library. Previously, I've done an initial screening of the whole DrugBank against a related coronavirus. So what I did was I took the top 1,000 or so compounds from that initial screen, and I screened that against this spike protein. Thirdly, we now looked at the stability I screened against the HACE2. Excuse me. Now I looked at the stability of the drug and the HACE2 receptor or enzyme complex to see how stable that interaction is. Then we moved on to see, having docked the drug on the HCE2, does it limit the level of interaction with the spike protein? So we had to find a way to quantify that and to show that the presence of the drug that was docked on the HACE2 limited that interaction. And of course, finally, we looked at the effect on the structural stability of the whole complex, looking at the HCE2, the docked compound or drug, and the spike protein coming into that interaction. And finally, we also looked at the current indication for the drugs that we found that are most likely to disrupt that interaction between the spike protein and the HACE2. So in terms of results, this is what we got. So we were able to get an average structure. And the reason we needed to do that was because proteins are very flexible. And this poses a problem when you are doing a docking study for you to actually get the most likely state of the protein that you want to dock to on. So we did a molecular dynamics simulation, and from that we got an average structure from the dynamic trajectories from the frames or the various conformation we got from the different frames of the simulation, and we were able to kind of calculate an average structure that we think is most likely that the ligand will encounter when it comes in contact with especially with the HCE2 onto which we want to dock some of the drugs within the DrugBank library. Now, after the docking, what we found on this graph shows the binding energy and some of the top scoring compounds that we found. BV2 showing very good binding interaction with ACE2, and the rest of the compounds also showed very good interaction. So we showed the compounds. As you can see, they are very long. Most of them are peptides that were docked along the interacting surface of the HACE2. So assessing the stability of that docked drug on HCE2, we ran a molecular dynamics simulation, and what we observe, if you look at the RMSD plot against the number of frames that we did for the simulation, what we saw was that in blue is the HACE2 complex to the drug, and in red is the HACE2 alone. That the complex of the drug and HCE2 are slightly lower RMSD compared to the ACE2 alone, HACE2 alone, suggesting that the complex is slightly more stable than the enzyme alone. And if you look at on the second plot here, which shows the number of hydrogen bonds that is formed between the drug and HACE2 along the trajectory or the frames of the simulation. And what we observe, you could see that the fluctuation of the number of hydrogen bonds formed from 14 up to almost 13 in number. And then this high fluctuation, of course, can be attributed to the flexibility of the ligand being, I think in this case, it was a decapeptide that was assessed for stability of interaction in this case. Okay, in terms of quantifying the effect of the DUCT compound on the protein-protein interaction between the spike protein and the HACE2. So we quantified that interaction in the presence and the absence of the drug. And what you see on this plot is the binding energy and the poses. So for in red shows the plot of the complex of the spike protein with the DUCT drug and the HACE2, and the blue line shows the interaction between the spike protein and HACE2 without the drug in between. And you can clearly see that without the drug in between, there's a stronger interaction between the two proteins, even across all the poses that were simulated. And in the presence of the drug, there's a significant reduction in the interaction between the two proteins, showing we can also conclude from this that the presence of that particular drug limited the interaction between the spike protein and the human receptor. On this graph, we now look at the stability of that complex. Now, looking at the spike protein, the DUCT compound, and HACE2, and on one hand, and the spike protein and HACE2 alone on the other hand. So on the top plot here shows the RMSD of the interacting surface for HACE2 in the presence of and the absence of the DUCT drug, which is BV2. So in the presence is the red one, and in the absence of the drug, you can see that. And C shows the plot of RMSD interaction surface for the spike protein in the presence and the absence of the drug. So if you look at the HACE2, you see a lot of movement. Now, that's what we observe, a lot of movement in terms of the flexibility. However, in the absence, which is the navy blue, of the drug, this seems to be a bit more stable. So we see this is a significant difference. And we also saw the same thing in the spike protein. So one of the explanations we gave to this was the fact that in this case, the drug seems to be disrupting, if you put them all together, the presence of the drug seems to be disrupting the complex of the spike protein and the HACE2, in the sense that it prevents that interaction, as we have seen in the previous slide, but also it prevents that interaction from settling down, as it were. So, and it kind of becomes an hindrance, a steric hindrance to that interaction. So there seems to be a lot of flexibility. If you look at the video, when they're coming together, the flexibility is a lot more because of that presence of that DUCT compound. And when we look at a closer look at the amino acids, especially looking at the interacting surface of the complex, you see that on the interacting surface, most of the amino acids on the interacting surface are constantly fluctuating. So I highlighted the interacting surface here. You can see the residues on that interacting surface showing a lot of fluctuation when in the presence of the DUCT drug. So when we looked at, so we've identified about 5 of them. drugs that should very good ability to inhibit that interaction, and most of them were peptides. And we went back to DrugBank and see what are these drugs used for. And one of the ones that we saw, SAR9 met all to 11 substance P, is a neurokinin receptor agonist. It's an analog of the naturally occurring neuropeptide called substance P. Substance P is a neurotransmitter for pain, and it's an investigational drug. And a further search revealed that the fact that this particular drug has been investigated for respiratory distress syndrome and also for viral infection. So that was quite an interesting find that this is also already been investigated, and we've been able to show at the molecular level that that this compound might also be beneficial in the related viral infection that causes depression in the respiratory system. The BV2, it's a cholera endotoxin subunit B, and when we look at the gene ontology term, we found out that that particular peptide, it binds to old cell surfaces, and it interacts selectively, non-covalently with the surface of most cells within the human body, and that opens a new door in terms of looking at a peptide inhibitor that can prevent fusion between the human cell and a possible, in this case, a virus from getting into that particular human cell. All right, so let me conclude that with some current related studies that we're doing. Of course, at the midst of the pandemic, we had a lot of variants that came out, and because we know from this study that I just described that peptides might be good inhibitors, so we started to look at these peptides and how they bind to the different, if you think about the receptor binding domain of the spike protein, and see is it possible to get something that binds to that and actually limit its interaction with HACE2. So we can reverse in this case, but we've not reported this particular study. Another study that I'm doing with a collaborator here, Dr. Bertram and D-World in my university, is looking at E-proteins, and the E-proteins have been implicated as a virulent determinant in SARS-CoV coronaviruses, and we are trying to see how we can also explore the drug bank to see if we can find potential inhibitor to this particular protein, particularly preventing it from its function, especially the PMB section of the protein, and limiting its function in terms of interacting with the pulse in human. And finally, we're also trying to explore the drug bank as a way to repurpose drug for to be used as alpha glucosidase inhibitor. Currently, we have acarbose and what's the other one? It's meglitine as an inhibitor of this particular enzyme, and this enzyme it's very important in if you're able to inhibit that enzyme to prevent postprandial hyperglycemia in diabetic patients. So we're applying both machine learning and molecular docking strategies to see if we can get substances or drugs within the drug bank that we can use as an inhibitor for this particular enzyme that is present within the intestine. So In conclusion, the drug bank, I believe, is a valuable toolbox for drug repurposing. I mean, we have a collection of successful drugs, and we can learn a lot from successful drugs. And in the era of AI, we can begin to think about AI-powered knowledge-based analytics that allows us to identify new indications for existing drugs. And I think there's quite a number of platforms that are already doing that, and we can do a lot more in that space in order to find and repurpose drugs for diseases that we are plagued with. Thank you so much. I really appreciate the opportunity to speak, and I will be looking forward to some valuable discussion, and also hopefully we will learn a lot from other people on this platform. Thank you.
Okay, so that was a fantastic presentation. We'll be moving into the Q&A period now. So again, feel free to throw questions into the chat bar. We'll be addressing them in the order in which we've received them. So we've had 2 questions during the presentation itself. I apologize in advance if I mispronounce anyone's name. I'll do my best. So we have one here from, I believe it's Nikhil. Suppose a person is taking an ACE2 inhibitor for a medical reason like blood pressure. Would that have any influence on the chances of COVID infection or RBD binding if there's already an ACE inhibitor in the bloodstream, as it were?
Okay, so thank you for that question. I appreciate it. So the simple answer to that will be that there was no significant difference. WHO actually did a study or reported a study, and they showed that there was no significant difference. Now, if you think about it logically and try to see, based on what I said in the presentation, what's happening there, it means that if you have an ACE2 inhibitor, for example, something that blocks, if you look at the RAS system that I described, angiotensin 2 for blood pressure, captopril, or any of those drugs, it means that the angiotensin, the level of angiotensin in the body is reduced. Now, if the level of angiotensin is reduced, there is no need for HACE2 to release its catalytic peptide to convert angiotensin 2 to angiotensin 1 and 7. So what has been reported in other paper or some other findings is that the low level of angiotensin 2 limits the expression of HACE2 in such patients. And if you don't have, remember that the virus needs the HACE2 to get access. So if you limit the expression, it means that on the cell surface of these individuals, there's less entry point, as it were, for the virus. And that in implication means that they will have a lower viral load in theory, and of course the morbidity they will experience will be lower. So that, I think, is for me trying to give a logical explanation to what was clinically observed, that there was no significant difference, as it were, for such people. I hope that answers your question, but you can, yeah.
Yeah, I think that's an excellent answer. It's always nice when there has been real in-depth studies of something like that from a clinical perspective. Okay, we have another question from, I believe it's Oguz. Not sure about that one, I apologize. But they ask, is the old function lost with drug repurposing, or are there situations where the old operation can be preserved just with new targets? I suppose operation here refers to therapeutic effect.
Yeah, indications. So yeah, very good question. And I think this is one of the limitations of drug repurposing. You can never have a perfect drug. I don't think it's possible. So, for example, I'm going to use the example that I gave in the presentation. Metformin is used for type 2 diabetes to reduce glucose level. Now, we found that for people, outpatients using that, it cannot reduces the effect of COVID-19 on them. Now, we don't know how, but somehow it's doing it. But it doesn't take away the effect of metformin on the glucose level in those individuals. So repurposing from one indication doesn't take away the primary indication. Now, and I think this is where dosing and pharmacokinetics comes in. So if you look at drug repurposing, one of the things you need to do, you need to repeat. It is suggested that you repeat phase 1 clinical trial in order to understand the efficacy for the new indication and the safety profile for the new indication. And that will also allow you, if you do the PK and pharmacokinetics, to know the dose. Now, now your new indication is your primary indication. The former indication becomes a side effect. So there needs to be a dose that will be titrated to minimize that side effect and now enhance the new indication that you're pushing with the repurposed drug. So that is where the trick is. For those that have been able to manage it, you might prescribe that drug at a different dose compared to when it is used for its primary purpose.
Yes, definitely. Okay, we have a question from one of the drug bankers in the audience. So Marisol asks, or she says, wonderful presentation. Is there a difference between approaching the E protein versus the spike protein interaction when researching COVID treatment alternatives?
Yeah, so the spike protein primarily is used as an ingress to get access to the cell that the virus wants to get into. The E protein has been linked, like I said, to virulence, determinants of virulence. So another thing that is also important to note is that if you look at the spike protein, there might be variability in the different coronaviruses in terms of the receptor binding domain. There might be slight differences, and we've already seen that because the mutants we saw started changing that particular domain in order to avoid vaccines, as it were, because it's kind of the easiest place to target if you want to build a vaccine. But the E protein may be more, let me think about it, the word I'm going to use, a better target to use because they are quite conserved amongst the coronaviruses. And if you have a conserved protein, it means that if you're able to find a drug for one virus, you might be able to use the same drug for the family, for the rest of the members of that family with that virus. So it kind of offers you a chance to get a broad spectrum antiviral, as it were. So I think it might be a better target to use, but it is problematic because every target must be druggable, and the druggability of the targets comes to play now when you look at the E protein versus the spike protein. Yeah, so that's I think it's my submission for that.
Perfect. Okay, the questions keep rolling in here. We have one from, I believe it's Keter, and they ask, is there a reason why you chose to do the docking of the drug molecule to the SARS-CoV-2 ACE2 complex rather than the SARS-CoV-2 protein alone?
Okay, so at that point, we were thinking the COVID virus is coming from outside. It needs to knock before it gets in to the cell. So, and the way it kind of knocks the door or kind of turns the knob of the door is through that HCA2. So think about it this way. So if you make the knob of the door very hot or very inaccessible, it's difficult for you to open the door. So that's the way we thought about it. And you also think about the fact that we want to give something to the host because we don't have access to the virus. The virus is coming from the outside. We want to give something to the host that acts as a barrier, technically, so that whatever is coming from the outside cannot get in. So the interaction surface of the host became something we wanted to take a look at and say, can we find something to block that interaction surface? And that acts as a fusion inhibitor for the virus that is coming in to fuse to that particular domain of that HCE2 in human. So that was why we focus on that. Yes, when we started having that changes in terms of the virus mutations, Omicron, Delta, and the rest of them, we began to think about, okay, fine. Is there a way to look at targeting the virus directly? I'm not so, in that case, it's more preventative in terms of, I don't want to talk about it, but there's so many other research that I did with some other people, really trying to see what does the receptor binding domain look like and what are the things that can inhibit that particular surface. But it was for a different intervention. But I don't want to mention that now. But yeah, that was why we chose the route to first protect the host from the virus that is coming into the host. That's why we chose docking into the HACE2. Thank you.
Yeah, I think that makes sense. Okay, so still waiting on other questions. I have a couple, actually, just while we wait for more questions from the audience. So I was curious by the finding that all of the sort of initial hits in your screen had this sort of extended conformation intended to be peptides. It made me wonder if there was some properties of the binding surface that you would have been able to quantify ahead of time that may have told you to expect a more extended conformation in the docking hits.
Yeah, so like I said, I first did an initial docking using the whole DrugBank. And from that, I could see when I look at the top hits, immediately I saw the kind of compounds that were ranked top there. So curious, I got curious and I went to the interacting surface that was crystallized in the protein data bank. And I looked at the HCE2 interacting surface, and you could see from the length of it, it's quite handful. And the spike protein coming together into that space, if you measure that, the length of that interaction surface, is quite long. If you think about a small molecule coming to that space. So there's no time to share that now, but if we look at the small molecules that's where we docked, you could see they occupy within the length of that surface. You see them, the different poses occupying different spaces within that space. And you can still see an interaction between the receptor binding domain and the HCE2. It's almost as if its presence doesn't make any difference. There's still enough space, enough surface for interaction. And I think that is why we tend to see that peptide-like. I mean, other people have also proved that most effective inhibitors that have been found have that long chain, flexible conformation in order to stop that interaction between the two interacting proteins.
Okay, it's very interesting, actually. So I have another question. This one is kind of switching gears just a little bit. But I know that one of the primary motivations behind drug repurposing is that it's generally less expensive than sort of starting de novo and doing the whole discovery process. But it's often the case that the compounds that are identified need to be tweaked a little bit to improve the binding. I was just wondering if you had any thoughts about the most efficient way to approach that particular issue, especially in more resource-constrained areas.
Very good question. The challenge now is once you tweak the compound, you form a new compound. And once you form a new compound, it's as good as de novo drug discovery. So you're going to have to go back all the way to your drug discovery and do all the preclinical studies before you can get to the clinical study. And so that is a big challenge. If you're lucky that you have sufficient interaction to bring about an inhibition that is clinically relevant, then you can proceed with that, depending on the dose that bring about that. So you can proceed with that drug, and most people, I don't know how many, I've not done a check to check how many drugs that were repurposed actually. And if you check most drugs that are repurposed, most physicians actually do that without really understanding the molecular mechanism behind it. It's basically based on observation. You just saw that this patient, while you are treating this, also manifested this particular phenotype. And you tend to say, okay, if I use it for that at a particular dose, I might be able to enhance that phenotype versus my primary indication. So that has been it. Now we're going back to understand why that is the case. And we're trying to now say, can we optimize it for another particular indication? For me, it's first of all defeats the purpose of drug repurposing. You defeated the purpose. If you cannot achieve drug repurpose with pharmacokinetic optimization, if you need to get into pharmacodynamics and compound optimization, then you've lost the primary initiative. Then it is as good as you. It's a very good start for. It's a very good start for de novo drug discovery. At least you have an understanding, then you can quickly optimize. Then you can move, and it offers you a very good advantage if you think about it. But I'm not sure the cost of preclinical development will change, and definitely you still have to do all the clinical development. So for me, we're excited about it, but at the same time, we know that it's not going to fit into all the diseases. Sometimes we have to rely on this serendipity. It happens to have this effect in some patients, and we go with it, provided it's safe. And if we can explain it, fine. If we cannot explain it, there's a lot of drugs out there without an understanding of their mechanism of action. But yeah, as much as possible, I think let's keep the initiative, the primary initiative, which is to find something that works for another disease and not really understanding how. If you want to understand how, very good. If you can find the pharmacokinetic parameter to make it work for another disease, beautiful. Then that's a win for us. Thank you.
Right, okay. So essentially having to tweak it is almost defeats the purpose of taking the repurposing approach is essentially the takeaway. Right,
Okay. Take it away.
Fantastic. Okay, so getting close to the end of the session, and I don't see any other audience questions. I have one last question to ask, and this is, I feel it's almost obligatory in this day and age, but you know, there's been an increasing number of AI or AI adjacent methods that have been coming out in sort of the drug discovery and repurposing landscape. I'm just curious if any of these new methodologies have got you thinking about a different way that you want to move forward with your research.
Yeah. Well, so like I said, one of the things we're also doing is to use this modern technology um, on the on the current uh, data sets we have in DrugBank. Um, DrugBank offers you, and I think this is, you can't take anything away from experience. You have a drug that has made it through and is sitting down there. You have a lot to learn from it. And machine learning, as it says, is enabling the machine to learn from what has succeeded. So from that perspective, it's something that I've particularly been looking at. Um, we've done it in so many, in some of our approaches to drug discovery, trying to see, for example, we look at a drug bank and say, in the initial characterization that I did, you find a particular cluster. If you map over that, the diseases, do we see a cluster linking up to a particular disease? If you think about that. So that's a very good data analytics to do. The other thing to do is think about the knowledge analytics that I spoke about, mapping each drug to the indication and mapping each drug to the side effects that has been reported. And if you're able to do that, if you imagine generating a network map, you can actually work backwards from that network map and be able to say, perhaps there's an hypothesis here that this particular drug may be useful in that. And that you can begin to now think about, how do we prove this hypothesis? But mining that data using the modern technology allows you to visualize what is possible and it generate hypothesis that I believe in the future we are setting out to test and see if we can yeah. Funny enough, one of the things that um, that we, when it comes to complementary medicine, especially traditional medicine in Africa, one of the things that has been a bane was that most of these herbal remedies, um, the owners or the people that discover them claim that they can work for multiple indications. We didn't believe it, of course, but if you think about it, it's possible for an herbal remedy that contains a milieu of compounds to be working for multiple indications. Yeah. So that is one thing I also been thinking about and say, if I look at an herbal remedy, see all the compounds in that and say, can I map each compound to a specific indication and generate a network map that becomes an analytic tool that we can use to generate knowledge and hypotheses. So there's countless ways we can use these modern analytical techniques in drug discovery. And I think it can only get better as we grow in the knowledge. Thank you.
Yes, I think that's everyone's hope, is that the field continues to move forward. Okay, so I think at this point, we're almost done, so we'll finish taking questions. And all that's left to do is, of course, to thank everyone for your attendance to this Powered by DrugBank academic webinar session. If you're interested in participating in a future session, if you're an academic researcher and you use DrugBank, and you'd like to share your research with the wider audience, you can contact DrugBank through LinkedIn, or you can contact our marketing individual responsible for this series, Kathleen. Her email is kathleen@drugbank.com. And of course, you can follow us on all of our social platforms for future updates and opportunities. And we'll also be making this particular session available on demand afterwards for anyone who signed up. You can find Professor Samuel Egieyeh on LinkedIn if you'd like to reach out and discuss any of the things we've talked about today further with him. And I'd like to particularly thank you, Dr. Egieyeh, for taking the time to come on. Yes, it was a fantastic presentation and a very, very good question and answer session. I think everyone learned a lot. So
Thank you so
Okay.
much. I appreciate it.
Yes, perfect. All right. Stay safe, everyone.
All right.